O să vă dau microfonul să vi-l puneți după ce fac introducerea. Welcome back, everyone. We hope you enjoy your meals, cigarettes, and you had time to network during the break. We will continue our conference with Professor Zoltan Balint, a senior medical physicist and professor of biophysics at the Faculty of Physics from Babeș-Bolyai University of Cluj-Napoca. With a strong background in cell biology and cell imaging, atomic force microscopy, biomedical imaging, and the development of computer-aided diagnosis solutions, his research currently focuses on biomedical data and image analysis to develop intelligent decision support system using an interdisciplinary approach. Today, Professor Zoltan Balint will inspire us with the workshop he prepared for us on interdisciplinary collaboration. Success!
Thank you very much for your kind introduction. I'm pleased to welcome you at this special session where I have to warn you because you have chosen to stay here in this dark room instead of having a walk outside because there is sunshine outside. So let's just put some light on here. And as you could hear, this is a workshop, so we'll have to work a little bit. But before that, let me show you why I consider that I'm qualified to talk to you about multidisciplinary networking. First, I started as a medical physicist here, then I did my PhD in biophysics, learning cell biology, biochemistry, and other methods. Then for 3 years, I was a postdoc and I was working with cells and tissue at the Medical University of Graz. Later on, I moved back to my original field as medical physicist, and I was a research group leader for 6 years at the Ludwig Boltzmann Institute for Lung Vascular Research, and still can talk about this part as well. But since 2016, we moved home, and I started my career already 10 years ago at our university. And how I imagined I would be working when I was a physicist, working with chemists, biologists, and medical doctors. So Michio Kaku said that physicist is an attempt to understand itself. So we would like to understand something. We have a question, and we want to answer to that question. So I would like you to think about a question, a scientific question. And as we have the groups, we'll discuss these questions, and we'll come to some kind of solution, to put together a team. Your task will be to put together a team to answer a scientific question, okay? And what we need, we need a blackboard, we have. We need some tools, physicists like lasers, you know, and some big guns, we can hit something, and then to come out with a solution. Here, that is nice, you can see, but how I thought I would be working as a physicist. So this is how I could imagine my work as an experimental scientist. How my relatives, friends would think that, oh, I'm working hard when I'm doing it. And what is it actually about? It's not like this, you know? You could see these solitude people. No. So actually, what we do, we work as a team. So for a team, one would need to have the question and to have the roles. Each of you will have to have some roles in a team. And yes, it's about a lot of work when it's about the theory and testing, of course. So we make theoretical part and experimental part. Now, as the intersection, we can start now, right? Please form the teams. You have team number 1, team number 2, team number 3, team number 4, and team number 5. It's fine? Please choose one of these topics. I would recommend you number 5, actually, but these are the topics I was involved in. So I might challenge you to think about a multidisciplinary approach for the detection of heart conditions, for example, MRI and blood biomarkers, dealing with patient samples. This could be one choice. Second choice, contrast agent development for MRI and CT as a basic research to put together some material which can be used as a contrast agent. Number 3, identification of cancer. Biomarkers, kind of a screening program when you go and provide blood tests and then have a blood test to analyze and to screen for the presence of cancer. 4 would be 3D models for medical applications, so CT, ultrasound, MRI, and so on and so forth. This is kind of an engineering task and applied science. It can be even reverse engineering. And 5, if the team decides, then the team can come up with another question, another scientific question. Your task will be to identify the roles in a team and whom do you need to perform the research to come to a conclusion. And nevertheless, as we are at the conference, Students for Students, at the Faculty of Chemistry, I would like you to think about the role of a chemist and a biochemist or a chemical engineer inside of the team. Is it clear? You have 5 minutes from now. You can choose number 5, of course, so come up with an idea. It's fine. If you need anything like material brainstorming, you know, you have some post-its, you have some papers, just put together. Three minutes passed. You have 1 more minute out of 5, I know. Please take your notes. At the end, you will have a 3-minute pitch to present at the very end, okay? I know 3 minutes are very long. Let's just keep it 1 minute then. Okay, so we have the 5 teams, right? Team number 1, did you succeed? Which topic did you choose? Tell me a number, please. Topic 3. Team 2? On topic. Team 3? Number? B. Okay. Team 4? Number 5? Team 5? Number 3? Boring, as one would say. Okay, so this is when we do not impose any of these topics. Nevertheless, I've been working with these topics, so I think and I do consider them fine. Don't forget, this is biomedical research. So when it's about biomedical research, that implies patients, data from patients, personal data. Then we have to talk about medical research. So this is the subject of ethical standards. And since the Declaration of Helsinki, all the human subject and the health and the right has to be maintained. So I had a personal experience when I had a division. I was leading the division of on a clinical research on patients with pulmonary hypertension, where we did develop dynamic computed tomography protocols to investigate the patients and to have to come up with a new automatic algorithm to analyze images. So this had a part of chemistry inside as well, because we had to deal with the contrast agent, which is administered to the patients. Generally, you know, in computed tomography, there will be around 40 or 60 milliliters of iodine containing solution administered intravenously. And what we had to do to develop a protocol to have a exact time point when we obtain the image to be able to analyze, for example, in this case, the lung vasculature. So as an idea from the image through the computers and to aid the reader, computer-aided diagnosis or computer-assisted diagnosis is about a possibility for the medical doctor to be used in the diagnostic process. And what is important in this case, it's not about replacing the medical doctor. It's about having some research behind to be able to have the medical doctors in the diagnosis. And this is what nowadays fashionably we call implication of automated algorithms and AIs. Nevertheless, when it's about biomedical research, then there are some phases, clinical phases, for example, to think about. Do you know about drug tests, how they are done in companies? It's active. Who knows how the clinical trials phase 1, 2, 3, and 4 are conducted? Okay, then let's talk about it a little bit. So there could be drug tests where pharma companies having a product, a biochemical product, which can be used as a, for example, as an agent against a condition, they are tested in several phases. And the other one, observational studies, they are researcher initiated. Now, please consider that you formed a research team and your study will be observational researcher initiated. It does not have a support from a pharma company. You have to put together all the people. You have to have the roles inside your team who will do what. Is it clear? So the next step will be to decide whether do you want animal or human participants, which type of material do you need, and who are the people who will be involved in your research. Okay? Just a short bracket here. Okay. Don't forget, when it's about human participants, then you need ethical approval and monitoring as well. So the same is true for animal experiments. Whenever you are involved any human or other living, you need to have ethical permissions. And there are the clinicaltrials.gov, which is a database. I can recommend for those who are doing biomedical research, because in this database, not all the studies are registered, but majority of the studies are registered. And there you have a possibility to search for ongoing any type of either company initiated or researcher initiated studies. And for example, this is how it looks like. When you do a search, then easily you can find and you can tag prior publication, ongoing clinical research. For example, I did enter, what else? SARS-CoV-2, which is a study which was initiated already at the very beginning of this infection. So what am I talking about? When you have to design a research on a specific topic, then beside identifying the roles, whom do you need in the team? The second one is to make your research on what is already done, what is published, and what is ongoing. As if it's published, it's not a research. It's clear? You have 7 minutes. Please identify the roles in the team and whom do you need as a specialist, and the research topic to break down how would you like to perform the research, what to use, human samples, animal samples, cells from a tissue bank, and so on and so forth. Go.
Ok, o să stau de fapt acolo și dacă cumva nu mergem, poate să...
Nu, sticul înăuntru. Și nu pot să merg schimb, dar o să fac schimb.
Acum acolo stau acolo și dacă cumva nu merge, am faceți un mix și eu dau slide.
Dar voi peste?
Asta nu merge pe televizor. Mai bine vă luați un box, nu?
Merge.
Dar nu se vede punctul.
Punctul nu se vede, nu, că e LCD și nu se vede deloc. E ok, că nu video ăsta și... Trebuie să merg să merg. Fiecare cu rolul lui. Am zis, trebuie să alegeți un rol. Pentru fiecare un rol.
Eu ne fi trimis
Nu avem nevoie de chimiști, avem nevoie de câte multe disciplinare, fiecare cu rolul lui.
Dar cum doctorul o să inițieze chestia asta?
Corect. Corect, de acord, doctorul o să inițieze, dar avem nevoie și de alte.
Doctorul inițiază atunci că vrea să descopere.
De aici vom continua imediat. Mersi. Super.
Nu cred că e corelație așa directă.
Super. Se poate obține și țesut deja recoltat. Dacă e cazul, de la biobank. Paranteză. Și atunci vă puteți gândi într-adevăr în direcția de a avea materia primă cu care lucrați la îndemână, ceva retrospectiv, după care ulterior se pot face și prospectiv. Super. Fly on the wall. You fly on the wall. Da, e total diferit. Ei, față de celelalte de pe da. E absolut super. Deci mă bucur că a venit invers. Am fi așteptat să fie ales fiecare separat, dar au ales acea schematică și abordare total diferită. O să iasă bine. E ok. Ați pus together? Aveți tot? I have to switch to English because then I will mess myself up with the languages. You have 2 more minutes. It's a simulation, it's a game, but still, we have to keep it quite short. Team number 1, do you have your material, what you would like to use? No, type of material.
Thinking of presenting organic molecule or molecule that can be transformed by microbial or protein that can be then by
Blood and tissue. Did I hear well? Okay. Team number 2. Team number 2. Do you have your primary material, what you would like to work on? Yeah, like for example, team number 1 has blood and tissue to work on for the research plan. What do you like to use for your research plan? Cell culture? Cancer cell line? Okay. Team number 3. What is your primary material you would like to use for your research? Tissue. Which type of tissue? Human or animal? Animal tissue. Nice. Team number 4. Human? Okay. So clinical trial. Clinical trial. Okay. Team number 5. Biofluid. Human. You see? The same topic and totally different approaches. I like it. So whenever we are talking about human samples, then we have to talk about clinical studies. And single and double-blinded clinical studies, they are conducted by pharma companies, for example, or researcher-initiated. And don't forget that there are sources of bias. There be the patient, how it's being treated, when do you get these samples, the clinical staff administering the treatment, or the doctor. And the masking can be at 4 stages: unblinded or open label, when everybody knows what's going on. So the patient knows what it receives, and all the participants are aware of the treatment. The second one, when testing drugs, there is the problem of bias. So that's why generally at the phase 1 and 2, they do single-blinded or single-masked studies where only the participant is unaware of the treatment. So the person who will be administering the treatment will know what this patient and the other patient will receive. There are, again, 2 more levels. Number three, double blind or double masked, when even the people who are administering the treatment or the biomolecule, they don't know who receives what. And the triple blind is the last stage where even there is a board which has no connection with the experimental part. They receive the data, they analyze it, and they open it later. And even the data analysts are all unaware. If you want to get know more about this topic, then I could recommend you this blinding: who, what, when, why, and how, Canadian Journal of Surgery, because it describes the clinical studies. And just to think about what could be a problem. A problem in a sense that what can happen. For example, in the late 80s, early 90s, there was a clinical study where there were the two arms well defined, and later on, the study had to be stopped because it was massively biased. Although it was a double-blinded study and some drug tests were made on patients receiving the medication and some arm with the placebo. And later on, they realized that systematically those who received placebo were treated differently by the personnel, by the medical personnel, and they didn't know why. Until it was totally blinded. All respected the protocol, and once they were discussing with the nurse, and she was saying that the substance is smelly, placebo not. It had a smell, and they knew which patient received the treatment and which patient received the placebo. This is a compromise where all the study, 10 years, was gone. Now, nowadays, there is a rule, and since the late 2000, you cannot give only placebo for a person in a clinical study. Clinical studies, you have to give to the person the medication, that's the baseline, and whenever they test another substance, they have to top it. And then you can test, or they can test, the pharma companies, for example. So, our second topic, biomedical imaging. What role can have in biomedical imaging for the physicists, for example? You know, as a biomedical physicist, you have to learn quite amount of biology. And our idea as a medical physicist is not about the above line, rather it's about the microscopes, the ultrasound machine, and other type of machines like the computed tomography and MRI and so on and so forth. So for your task now, we identified the material you need. We identified the team, right? Who does what? Now comes the third part. What instruments do you need? What do you need for your research? Do you need primary data? Do you need patient data? Do you have to collect patient data? The next step is, for 5 minutes, please discuss what type of measurements, what you need to put together to measure these samples. Okay? Please, go, 5 minutes. Nu am testat în real life, încă nu mai... Despre modele 3D și ce face.
De contrast, agent de
Da.
contrast.
Am
de
făcut
identificare. La idee, de identificare.
agentul de contrast este substanța care se dă la CT și VN pentru a vedea mai bine imagine. Și biomarkerul este ceva care provine din sânge, din lipid, este inerent pe persoană și, de exemplu, avem un proiect unde încercăm să vedem nivele crescute de ADN circulant în sânge. care este un martir a prezenței unei boli, în general cancerigene. Că de la apoptoză mai rămân fragmente de ADN în sânge și creșterea nivelului de concentrație de fragmente de ADN este un semnal că ceva nu este specific, este absolut specific. Și cu colegii studenți de ani avem nanoparticule care se leagă preferențial sau pot fi funcționalizate să se lege preferențial de ADN și cu microscopie putem cuantifica semnalul de la nanoparticule care se corelează cu concentrație SERS, Raman, adică, mă rog, clasic și avem cea de fluorescență ca și reporter, dar acolo deci pentru ADN. Maria, de exemplu, a funcționalizat nanoparticule, a fost de silica peste, stratul, acolo era problema mare, dacă e mult prea mare, o lume de chimică, și atunci trebuie redus ca să fie acoperit și să se leagă preferențial ADN-ul. Ideea a venit de la coloană, când e silica. Da. Invers. Înăuntru avem argintul și argintul la de semnalul SES și un strat foarte subțire de silica peste, unde ADN-ul se leagă preferențial și putem să avem probe, de exemplu, unde am arătat chiar de la fluorimetrie. Iar pentru a vedea dacă ADN-ul este unicatenal sau dublu catenal, dacă e distrus și cum e distrus, avem reporterul Cyber. Cyber este un agent fluorescent care se leagă preferențial de catena, dublu catenal. Și asta se folosește de către biologi, că de aia altfel să discutăm cu el. Și atunci ideea este agentul hotspot ca să am înțeles. Și când se fac conglomerate, atunci o să am o amplificare a semnalului și pot să le țărsi. Deci asta e doctoranda mea, lucrează acum. Da. Da, și atunci acolo și se poate face. Da, dar you have one more minute. Please summarize. Please, do you have a role in your team who takes notes? I mean, you have to have somebody who will take notes and put together, okay? This I forgot to tell you at the beginning, but obviously you have somebody who will take notes, right? Cred că știu care e problema. Undeva are sensul și direcția asta, că la calculator. Și atunci dacă stau de față, nu mai vreau. Și nu știu cum ar trebui să fac.
Lasă-mă schimba bateriile ieri.
Nu e baterie. E cumva setat undeva și este un timer și merge timerul și asta e problema. Adică e setarea instrumentului.
Oare, haideți să încercăm să putem să-l redeschidem.
Acum a funcționat. Deci asta era problema, că nu am înțeles care e problema. Și un fel de timer setting care apăs din greșeală, antrenor sau cine.
Da, normal.
Și când apăs,
Nu, nici cu timer, nici cu timer.
și zice că e ceva. Da, pe mine nu, de așa de mult, că oricum asta e important. Okay, so you have some results, right? Please tell me. Team number 1. Fluorescence. And the other one, please? Neutron decay. Okay. So you need at least 2 very costly instruments, right? And somebody who can operate these instruments. So it's not a plug-in play thing. It's kind of a expertise what you need here. Team number 2. This was said when we started at 13 o'clock that I know that I have 10 more minutes. But now, as we have the delay, we can have a little bit longer if you are in the game. Team number 2. NMR. Two. You have to select the two most important ones. Yeah, I agree with it. Sam and Tam. These are three. But I know that there are STEM as well. So we are talking about NMR spectroscopy for a method to, again, it's a costly method. And then you have to have a qualified personnel who can prepare the sample, who can make the measurements and analyze the results. SAM and STEM, scanning electron microscopy, transmission electron microscopy. You need the same qualified personnel. Team number 3. CT or MR scanner. Okay, this is animal tissue, so you need a micro CT or a micro MRI. So you cannot use a human CT or a human MRI. Nonetheless, for example, we put some bones in the human MRI and it functioned very well. And in a human CT, for example, but nevertheless, in this case, you have to find a micro CT or an MRI. You know that, for example, here in the Inspire building, we have human MRI, very high performance, and micro MRI, which you can use it, 11 Tesla MRI. So actually, you are lucky because you don't have, you just have to cross the street and you have the, your device. What else? Coloration of the cell. What would you use to identify color in a cell? Some biomarker, but what would be the instrument, the readout? UV spectroscopy, right. It's a simple technique, UV spectroscopy. You have to prepare your sample, you put it in, you get the spectra, and you can analyze it. So this is the first technique which it does not cost that much because we are planning the budget at the end as well, right? Not today. It will be your homework. Team number 4. CT MRI, but it's a clinical trial, so you need a human CT and a human MRI. There you need two expertise, a person who can operate it and the medical doctor who will give the result, because we as a physicist, medical physicist and or biochemists, we are not allowed to give a result on the CT and MRI scans, since here you need the medical expertise. Okay, what's the second one? Q&A. This is what you said. Did I understood well? It's a questionnaire, a QA, which you put together to analyze the status of the patient. Here you need an expertise for, you don't need material because it's a paper but a discussion, but you need an expertise, a person who who has the right to talk with the patient, who has the right to talk with the patients and to get the results. Team number 5. That's all? Really? I'm not sure that's all, because, for example, when you are using a spectrometer, a surface spectrometer, and some nanoparticles, you need a second one to provide, am I right? UV spectrometer to provide the quality control for the nanoparticles and for your sample. But nevertheless, the surface spectrometer, so the surface enhanced Raman spectrometry, it's a very good technique to identify nanoparticle related and nanoparticles which can bind, for example, material, a specific one from a biofluid. Okay. I'm interested in medical imaging and biomedical imaging, and exactly this is what I expected. Can you see this here? I could make it bigger, and it's obvious, but I didn't want it to, because this is an image. Do you agree with? And then what's this? This is the same image. Ah, no, this is what the computer sees. This is what we see, and this is what the computer sees when it's about an image. So when we are talking about CT, MRI, or microscopy techniques, any type of microscopy techniques, we receive images and we do an analysis. Image analysis means that the computer will be dealing with numbers, X and Y columns, some numbers. Each number is a code for a color. And you can easily identify there are two wires here, here and there. And then you can see this is wire 1, this is wire 2. I did color it to see better that this transition, small numbers means black, large numbers means whiter. And these differences, what we are searching for in a CT, MRI, or any type of image-based diagnosis in radiology, to find this, the delineation of the object. What radiologists do, they do delineate objects on an image to find out. And why do we need then medical doctors? By the way, chemical engineer in the team and another chemical engineer and some medical doctors. You have your team, right? This I forgot to ask. And other people. I had the pleasure to work with live cell imaging fluorescence microscopy, and I got the question from my former colleagues from the Biophysics Institute doing basic research that how could we understand the function of an animal or human or other life forms when you even don't understand how a cell functions or even a small protein. So what was the main problem? Working in a multidisciplinary environment, I couldn't understand a word what they were talking about at the beginning because there is a code of the language, you know? Certainly. I mean, in the medical field, in the biomedical field, they are talking about BPA, okay, RPA, LPA. When it's about radiology, then it's obvious for them, it's the right pulmonary artery, left pulmonary artery, but they were talking code, LPA, RPA, and so on and so forth, and I couldn't understand what they are talking about. So, is there a biochemist guide for the medical language? No, but I would love to have it, okay? And probably, if you enter the biomedical research field, at a certain time point, it will be for you as well. Very important to code the language. I functioned in the last 10 years as a translator between my colleagues, medical doctors, specialists, physicists, and computer scientists because we are developing one-to-one automated solutions from CT and MRI images for the diagnosis. And the problem at the beginning was that they were talking about the same topic, the same thing, and they couldn't understand each other. So in your team, it's okay if you have a biomedical physicist or anybody else who would take the translator role to coordinate this. Second point, biomolecular targets. This is what you have chosen. Topic number 3, right? Some biomolecules which can function as a biomarker. And what was very interesting, we had a study on patients, and in this study, we arrived to a conclusion. We received the data, and I was very happy with Philip, my PhD candidate student, that we find a very nice solution for one parameter as a biomarker in the blood. And when this value was above 120, then it was perfectly separating the not that ill, I mean, condition number 1, lung disease, and condition number 2, lung disease stages. And we went to the professor and showed them, yeah, this is what we have. It's wonderful. It's above 120 and it makes a perfect cutoff. And he started with this one. Perfect biomarker does not exist. One biomarker parameter, it's good for nothing. And nevertheless, if this parameter is above 100, then I have to be with the patient in the operating theater already because it's very ill. So we did not patent this biomarker, by the way. Nevertheless, the cutoff value for biomarkers, you know, right? You participated at blood tests anywhere, and then you receive an interval, a cutoff interval, of your value. If it's outside the interval, it's a condition. So bottom line, it could be mathematically significant, but you have to check for relevance, for medical relevance and accessibility. And another story, and with this one I will stop here and I will let you work on your project. The other story is biochemist. I was just collecting those biochemists whom I encountered. And she's Angela. She was working for 7 years as a senior developer for Bayer in Germany. And then she said that it was enough for her. And since then, she's working at the Vienna Patent Office, and she's in charge with biomedical topics. So a possibility for a biochemist or a chemist, actually, or a chemical engineer, is not only to work in the domain and to enter a company, which they are all hiring people like you, but also there is a possibility at a certain level to become a senior researcher, a group leader, and or to choose another path, right? For example, studying a little bit of law, and then to become a professional who will re-evaluate patents in a patent office and give an expertise on these points. It's your time to go on with the next task. Okay? You have the team members. We have the topic, the samples, the techniques, timeline. Please provide a sequence, a timeline. How many patients? How many dishes? How many animals? How many people volunteer in the study you need? And how you will collect the samples and how you will arrive to a experimental solution. You have 5 minutes. Nu știu ce urmează. Nu, în general, și asta le zic, deci, și la și fiecare slide, știu cât să pasă. Dar eu nu, că vine parte din Cluj acum. Și... Da. Asta. Numai că asta mai puțin.
Da, îmi dau seama.
Dar eu cum? Da, nu, am văzut. Că am fost și aici, și am făcut. Ok, 10 minute. Bun.
Lumea chiar...
Eu pot să vorbesc încă jumătate de oră, că am pregătit să iei, acum nu este angajat de este. Acum am gândit că să fie ridici. Decât pe 3 minute, trebuie să zic o experiență.
Exact, da.
Nu că experiențe ce au făcut. Aici am și o întrebare.
Da.
Mai degrabă, să zică ce au făcut și ce au făcut.
În task-ul ăsta?
Au făcut.
Da. Păi, oricum, cam ce au făcut au zis deja și decât să prezinte acest lucru, am gândit ce s-a părut mai bine de chestia asta.
Ce mai întâi?
Da, bun, perfect. Ce mi se pare la fel?
Ce mesaj, aș mai fi avut o tematică la final, dar asta putem sări. Nu putem sări. Tocmai de aia nu putem sări, că o să fie probleme aici. Asta vreau să le arăt. Asta vreau, neapărat. Că excluded și cum am ajuns. Deci o să zic asta.
Da, oricum, mie personal îmi place foarte mult.
Foarte activ. Deci eu am învățat de la Simona Motorneanu și de la the food recomanda Babson College.
Ok.
Entrepreneurship Formation. Foarte recent am trecut în test, am înrolat la ei într-un program și acum 2 ani aș fi fost să socotiți, să faceți și să capăți și mai departe. Deci cum ne-am schimbat, nu știu ce trebuie să fac. Total diferit. Deci, No, asta e singura problemă, că asta de 3 ore.
Da, da.
Nu, e ok, e ok, ajungem unde vreau. Mai am o linie. Absolut. Absolut. Nu, pe 10 minute. Deci, 10 minute și terminăm. Cine consideră că ar mai avea nevoie de timp? Un minut. You can negotiate with me. Sorry. If I speak in Romanian, I switch immediately to English and so on and so forth. So if you need more time, you have 1 more minute. Timeline, please.
So we're thinking of 3 months period for testing, and we have around 8 samples to taking. We are still doing it the whole thing.
So what we could hear here, you will do a proof of concept study, a POC study, classical, where you need 18 samples, and you will 80, 80. Thank you. 80 samples and 3 to 6 months testing period.
Yeah, and we are planning on integrating our biomarker and also finding all the
In 6 months. Very optimistic. I do like it. I do like it. This is what we had as well. We said that in 6 months we will test in the south in this project. We'll test like in 6 months a material which will be synthesized by our colleagues. But after 6 months they arrive to the synthesis to finalize the synthesis. So I do agree that 6 months could be good if you have the material and you know what you're testing. Cool. Then it's fine. I understand. What else? That's it. Team number 2. Team number 2. Synthesis, 12 months synthesis, okay? Well months cells trial. 12 months animal testing. Beautiful. Yeah, when it's about any type of marker and clinical trials, then you have to think of 5 to 10 years to be tested. But for that one, you need a company. So here, I would suggest that we could stop because you have like basic science and, okay, applied science in a proof of concept level at a cancer cell line, and you can arrive in 3 years, like, okay, here we said 4 years in this project, to arrive to a conclusion in 4 years, having enrolled patients here at the hospital. We had a multidisciplinary team, having enrolled patients, and we planned with 150 patients. This project was from 2016 to 2020, and what was the problem? The problems were the following. We had echocardiography and MRI, and suddenly only out of 123, 99 arrived to the database because of various reasons. When you plan an MRI on patients, you have to think of claustrophobia, you have to patient compliance, and all the other type of problems which can arise in a human clinical trial. That's pretty much risky. Nevertheless, and don't mention 8th of March 2020, you know, when they locked it down. We arrived to stop our experiment where it was. So nevertheless, we managed to have 36 controls and 63 patients with atrial fibrillation to be able to evaluate. Team number 3, your timeline, please. 6 months trials. 3 months, okay. 12 months tissue test. 6 months, and my blackboard is over. Nice, very nice. So we could hear a total of 3.5 years, if I'm right, right? I did add up what you said. So it ended up with me 3.5 years. So it's kind of a classical big project time spent, like we could hear in the not a proof of concept, like in the first team. The second team proposed a 3 year. Here we have a 3.5 year, including phase where you analyze the results and where you don't do any more experiments. This is what I said in our topic. That was a COVID lockdown, so we had the data. We had nothing else to do to analyze, but it's very nice. And then don't forget that when you prepare what we are doing here, you will see at the end, we prepare actually a project proposal for your idea. Number 4. 5 years. 2 sessions separately. First session. I got you. So per you have like 10 sessions, 2 per year. You have inclusion criteria. Fantastic. This we didn't talk about yet. So for any type of human trial, you need some inclusion criteria and exclusion criteria. You have to establish before handing in for the ethical commission your proposal that you would like to do this and that research. You have to prepare the protocol, what we are doing right now, and you have to include, even for animal studies as well, a justification, and for human studies, the PIC, first patient-informed consent, and you have to have inclusion and exclusion criteria to avoid any type of bias. Because, you know what? It's simple. I could show you as well that we had some controls and some patients. Guess the average age of atrial fibrillation patients. Say me a number. 40? 56? 60. And when about control, then we said that we could recruit people to do an echocardiography and do an MRI and do some cardiac testings. What was the average age of the people who signed up as volunteers? Your age. And then, if you do this comparison, then that's a problem because you are comparing apple with pear, okay? And your result is not validated. Therefore, that was the initial phase. Therefore, we had to recruit age and sex matched patients. So any type of clinical trial has to have inclusion criteria, exclusion criteria, and if possible, age and sex matching, that you assure that you are comparing apple with apple. Optimistic. We were as well, we said 200. Yes. 300. So this is again a problem. What is the problem? You have to analyze your surrounding, whether is it accessible. If it's about a condition, a rare disease, to find 300, and you need the whole continent to find 300. When it's about diabetes mellitus or hypertension, then you are fine. You just go on the street and you collect 300 people in our surrounding, okay? So when you have your question, you have to think about feasibility. The feasibility in this case would be Like, is it possible that in 2 years you get 300 patients, because probably you have to do a follow-up as well for your question. Really nice. And you plan 1 year then. 100 samples, 1 year. I have a question. What if after 6 months your fridge broke down? What if after 3 months your computer will say empty file data loss? What if, where did we arrive right now? We arrived to the last point, and this is my conclusion before ending the session. The last point is the risk analysis. You have to do a thorough risk analysis for your study, because you have to analyze all the points which could go wrong, and we know for sure that they will go wrong. So, therefore, in this analysis, one has to provide solutions for the possible problems and think about, this will be a homework because we have 3 more minutes. To sum it up, I am a medical physicist, and just to make a little bit of commercial, on the 7th of November is the International Day of Medical Physics every year. So medical physicists, they do study physics because we need to understand the radiation and its interaction with the biological samples. When it's about medical devices, then the medical physicist works in the hospital and will assure the quality control of the instruments and that they function properly. And we have to study some biology and chemistry as well, because we are dealing with molecules, living organism, contrast agent, and nevertheless a little bit of computer science, because we need image and data processing. So this is how we arrive to the end of our discussion, where I think you realized at the end, which I didn't say you at the beginning, we put together a project plan. We just have to write it and send it in for some approval. I mean, we need a budget as well. But we went through the steps of identifying the problem, testing whether it's relevant or not, analyzing what you need as a sample for the project, what is the technique you are gonna use, and what expertise you are needing your team, the timeline of the project, and from now on you need just one more tiny risk assessment, beside one more tiny little thing to find somebody who will finance this project and then to have some good results. Thank you for your attention. Questions and reflections.
Thank you, Professor, for your delightful insight, and congratulations on all of your work. Now we will open the floor for the last time.
And if I may start with the questions, because this is a kind of upside down role, so I would like to have each team to answer one question. What was the hardest thing during the discussion with the peers to decide on? Which part you spend the most time brainstorming?
I think for us was how we're gonna test the if our compound binds to itself, because using isotope as a case, it we have to think of how we can bring it to like a commercial amount of that
Mhm.
that's the most part. I think
Thank you. What was the easiest to convey on out of these topics? The easiest. How long did it take? Right. Nice. Who was the most active in the team? Was it a natural selection or you accepted this? Oh, thank you. Who brought the best idea? Who brought the best idea? Team effort. Thank you. That's cool. That's cool. Yes? How do you feel now? Amazing? Thank you. Enjoy your day and see you at the posters then. Thank you. Sorry.
Just the last announcement. We will continue with the second part of the gospel section today until 3 p.m. when we will return back to 1977 for the gospel ceremony. Enjoy.